Okay, so a very good evening to all of you. Please just let me know through this technical poll whether I'm clearly audible and visible to you or not. Okay, I need your response to know that I'm audible and visible to you or not. Davangiri? Okay. So let's start our today's session. In the previous sessions, we have started. Okay, in the previous session, we have started with the chapters. Okay, and we have covered the two genetics chapter, that is principle of inheritance, molecular basis of inheritance, and the biotechnology principle and processes. In the previous classes, today we are going to discuss the previous year questions of the chapter biotechnology and its application. As you know, this genetic unit is having a high weightage from the 12th class. So this is the last chapter of the genetics, or we can say the biotechnology. So please do all the questions today very carefully, right? And these all are the previous year questions, so it will help you to understand that how this chapter is important and from which part you will get the most of the questions and in which way you will get the questions. Okay, so it will only help you to understand. So on that note, let's start with our first question. So the first question which is given here is came from the NEET 2020, and the first question says the BT cotton variety that was developed by the introduction of toxin gene in the Bacillus thuringiensis, that is BT, is resistant to. Okay, you have to give your answer in a poll, but before launching the poll, let me read out all the options. Your option A is insect pest, B fungal diseases, C plants nematode, and D insect predators. And those who are not able to give their answer in poll, and for those who are this poll is not visible, can give their answer in chat box. That the question is, okay, question is already there. So the BT cotton variety, which is developed by the introduction of a gene, a toxic gene from the Bacillus thuringiensis bacteria, right? That is why we are calling it a BT cotton, right? So this BT cotton is resistant to why we have introduced this gene in a plants. Okay, so you have to give the answer. Okay, so Devangiri, you have given in the chat box that it's option A. Okay, that's right. It is option A. Okay, so we have seen this Bacillus thuringiensis is having a gene that is known as cry gene, right? Now this cry gene is responsible, sorry, this cry gene is responsible for production of a toxin substance in a crystalline form, right? These crystalline structures are protoxins, right? This cry gene produces protoxins, and these protoxins, that means protoxin, it is inactive in nature. So these protoxins get activated when they get the alkaline pH, okay? Alkaline pH. And alkaline pH is only possible when the some part of the plant, when the plant is eaten by the insect, right? When the insect eats the part of a plant, that toxin granules enters into the gut of the insect, right? And the gut of the insect is having the alkaline pH. The gut of the insect is having alkaline pH. So due to that alkaline pH, as I said, the activation of protoxin only takes place in an alkaline pH. So that get converted into the activated toxin. And this activated toxin causes the lysis and inflammation. Lysis and inflammation in the gut of insect, right? Due to which pores appear on the gut, and when the pores appear in the gut of an insect, the insect will automatically die because of starvation, right? When there is gut inflammation is there, it will causing a lot of pain. So that lot of pain always leads to the hunger, right? Where the insect would not be able to engulf any of the stomach, any of the thing. So due to which that insect will ultimately die because of starvation, right? So this is how the gene work, and these pests are corn borer. These pests were corn borer and cotton bollworm, right? Okay. So let's move to the second question. The second question here is what triggers activation of protoxin to active toxin by Bacillus thuringiensis in fall form, right? Your option A, acidic pH, option B, body temperature, option C, moist surface of the midgut, and option D, alkaline pH. Now you have to give your answer in this poll, and those who are unable to give the answer in poll can give their answer in chat box. Okay, so again I'm repeating your question. The question says, what triggers activation of protoxin to active toxin of Bacillus thuringiensis in bullworm? Bullworm is the insect, right? It is an insect pest. In this, the inactive protein which is present in BT cotton, BT brinjal, right? They get activated. Okay, so very good. So okay, one of you have given answer option B and one of you have given option D. Okay, let's check it. Right. So as I have already discussed, beta, that these Bacillus thuringiensis bacteria are having a gene that is known as crygene, right? These cryogenes are responsible for formation of a toxin. That is a protoxin because that toxin substance is not in an active form. Whenever the insect bites the plant, these toxic granules enter inside the mouth of the insect, right? When these crystals of toxin get in contact with the alkaline pH of the gut of an insect, they get activated, right? They get converted into the active toxin. And this active toxin causes the lysis and inflammation of gut of an insect, due to which the insect ultimately die because of starvation. Okay, so this is just we have discussed. So the answer to this question is option D, not B. Okay, so body temperature not matters. The main important thing is the alkaline pH. Okay, is it clear to everyone now? If is it clear, please use the thumbs up reaction. Okay. So your next question says, which of the following is true for golden rice? Now here we are discussing the golden rice that is also yellow in color. Okay. Usually the rice are white in color, but here the rice is yellow in color or goldenish in color. So your options A says it has yellow grains because of a gene introduced from a primitive variety of rice, right? Question option B says it is a vitamin A enriched with a gene from daffodil. Option C says it is a pest resistant with a gene from Bacillus thuringiensis. And option D says it is a drought tolerant developing from the Agrobacterium vector. Now I am launching the poll. Please give your answer in this poll. So you have to find out why the color of the golden rice is yellow and or the correct statement regarding the golden rice. Okay. Okay, 5 seconds more. After 5 seconds, I'll close the poll. Okay. So all of you have chosen for option B. Yes, that is the correct answer, because we know the rice is a staple food in India, right? But it is deficient of vitamin A, due to which many of the peoples are having the problem of night blindness, which is caused by the deficiency of vitamin A. So to enrich the staple food so that we'll not get the deficiency, we have introduced the vitamin A synthesizing gene, right? The beta carotene gene. The gene is beta carotene gene from a daffodil into the rice, right? So that just by taking the rice, the appropriate amount of rice, we are able to eliminate the deficiency of the vitamin A and cure the night blindness. Okay. So next question, fourth is, in RNAi, the genes are silenced using. RNAi is RNA interference technique. In RNA interference technique, the genes are silenced using the double-stranded RNA, single-stranded DNA, single-stranded RNA, and double-stranded DNA. You have to give your answer through this poll. Everyone has to participate in poll so that you get to know that you are giving the correct answer or not. Davangiri, try to answer in a poll, beta. Okay? Okay, 5 more seconds for the poll. JNB UK, you also have to answer in this. Okay. Okay, so as Davangiri has already answered, it's option A, right? It is option A. Because what is happening here? We are making a complementary strand of RNA to our complement to our RNA, right? For that, we have to introduce another RNA, right? So whenever we introduce, right, the double-stranded DNA, it will cause the copy. It will make the copy of two RNA. One is silenced and one is sense, and other is nonsense RNA. So these are DNA fragments, these RNA fragments get sliced up and form the siRNA, right? siRNA. Now these siRNA, this is our double-stranded RNA, okay? When it gets sliced up inside the cell, it is forming the small, very small fragments of RNA, right? These small fragments of RNA is known as siRNA. Now this siRNA is capable, it is capable to form or get joined with our enzyme, or we can say the protein. So when this siRNA binds to the protein, it forms a complex which is degenerated by the cell mechanism and causes the inactivation of protein. So this is how, by introducing the RNA, right, by giving the silenced RNA, we are able to inactivating the protein. So your question number 5, which part of the tobacco plant is infected by the Meloidigan incognita? Everyone knows this Meloidigan incognita is a nematode parasite, right? It is a nematode parasite, due to which the production, or we can say the yield of the tobacco plant decreased a lot, right? So you have to tell me this infection, or we can say this parasitic nematode is found in which part of the plant, right? Okay, everyone can give your answer in chat box. Right, that is answer B, roots. We have studied, it is also given in our NCERT, that the tobacco plant roots were having some nodules-like structure, right? And those nodules were carrying the parasite nematode. And due to the presence of that nematode, the tobacco plant was not getting proper nutrient and it is affecting the yield of the plant, right? So next we have the question. It says golden rice is a genetically modified crop plant where the incorporated gene is meant for the biosynthesis of option A, omega-3, option B, vitamin E, option C, vitamin B, and option D, vitamin C. Now you have to give your answer in chat box, and those who are not able to give their answer in chat box can give their answer in this poll. We have just discussed about it that we are making the golden rice, right? We are making the golden rice to eliminate. We are making the golden rice to eliminate the deficiency of. Okay. So we were using the beta carotene gene. Here we have introduced the beta carotene gene from the daffodil. And this beta carotene gene is responsible for the formation of vitamin A. Okay, just tell me the name of the process which we are doing. When we enrich some plant for a particular nutrient, when we enrich a plant for a particular nutrient, that process is known as okay, what do you say? There is a term we used in biotechnology while making the plant enriched in a particular nutrients and minerals. Can anyone give me the answer? Yeah, that process is known as biofortification. Biofortification. In this process, we are making a particular plant, right? We are making a particular plant enriched in a particular nutrient. So here we are making the rice enriched with vitamin A. So this process is biofortification, which comes under the biotechnology. Okay. Or we can say the biofortification is an application of biotechnology. So your next question is in Bt cotton. The Bt toxin present in the plant tissue as protoxin is converted into the active toxin due to A, action of gut microorganism, B, presence of conversion factor in insect gut, C, alkaline pH of the insect gut, and D, acidic pH of the insect gut. Okay, give your answer in poll. Very good. Okay, so 5 seconds are left. After 5 seconds, I will close the poll. Okay, so as you know, everyone has given the answer C. And we have already discussed this concept in the previous questions 2-3 times, that in alkaline pH, only this protoxin get converted into the active toxin. Okay. Okay, next. The crops engineered for glyphosate are resistant or tolerant to. Now we are talking about the crops which are resistant to the glyphosate, right? Glyphosate is a chemical compound which restricts the growth of an plant, right? So option A, insect, B, herbicide, C, fungi, and D, bacteria. Now you have to find out your answer. Okay, now you can feed your answer in this poll. That glyphosate, the crop engineered for glyphosate are resistant or tolerant to Which out of these four, insects, herbicides, fungicides, or bacteria? 5 seconds or more. You have to give your answer in this poll. Davangri, you have not given your answer. Okay, so I'm closing the poll. Okay, so as none of you have given the answer, see, the glyphosate is a chemical substance which is known as herbicide, which is used to kill the weeds. Okay, we can say it is also a weedicide. The glyphosate is a weedicide, right? What happens in a plant? Okay, in plants, there is a pathway that is known as shikmic pathway, which is responsible for the growth, which is responsible for the production of auxin hormone. But when this glyphosate acts, that particular enzyme which is responsible for the formation of auxin get completely deactivated. If that enzyme get deactivated, auxin formation will not take place, right? If glyphosate is sprayed on the plant, the auxin formation will not takes place. And if the auxin will not produce, as you know, the auxin is the major growth hormone in a plant. So the growth of the plant restrict and plant will die. So this is how the glyphosate is used as a weedicide. But when we engineer the crop, we are making the plants resistant to this herbicide, so that when the weeds grow in our crop field, then the glyphosate only kill the weeds, and they will not harm our plants or the crop, because the crop is also a plant, right? It is also the what? It is also a plant. It is also a dicotyledonous plant, right? So the answer is B. Your next question says, which of the following BT crop is being grown in India by the farm? Option: brinjal, soybean, maize, and cotton. You have here we are talking about the BT crop. So out of these four, which is practiced in India, your poll is here. You have to give your answer. Option A says brinjal, B soybean, C maize, and D cotton. Now everyone, please give your answer. Okay, 5 seconds left to close the poll. Okay. So that is BT cotton. BT cotton is the most popularly used by the farmers of the India, right? Which is BT derivative. Next question says the RNA interference involves. RNA interference involves A, synthesis of cDNA and RNA using reverse transcriptase. B, silencing of specific mRNA due to the complementary RNA. C, interference of RNA in synthesis of DNA. And D says synthesis of mRNA from DNA. Now you have to choose your option. Everyone, you have to participate in this. So what will be your correct answer? Yes, right, absolutely right. That is the silencing of specific mRNA due to the complementary RNA. We have studied that we are producing the double-stranded RNA, which is having the sense and the antisense code. Very good. So your next question is, consumption of which one of the following food can prevent the kind of blindness associated with vitamin A deficiency? Out of these given four plants, which is able to reduce or eliminate the vitamin A deficiency? A, flavor saver tomato, B, canola, C, golden rice, and D, BT brinjal. The poll is here. Please give your answer in this poll. We have to talk about the plant which is responsible for eliminating the deficiency of vitamin A. Okay, very good, everyone. As already everyone has given their answer, and they also have given the correct answer. So I appreciate you all for the correct answer. Yes, that is the golden rice, in which we have introduced the beta carotene gene to increase the amount of vitamin A. So here is your next question. The next question says, tobacco plants resistant to a nematode have been developed by the introduction of DNA that produces in the host cell. Option A says both sense and antisense RNA. B says a particular hormone. C says an antifeedant, and D says a toxin protein. So here is your poll. Give your answer through this poll, that the tobacco plant resistance to the nematode have been developed by the introduction of DNA, which produces. Okay, only 50% of you have given the answer. The rest, please also give your answer. The poll will last in 10 seconds. Everyone give your answer. Okay. So everyone has given the answer. Yes, that the correct answer is option A, both sense and antisense RNA. We have known that we have introduced the double-stranded DNA, which is responsible for the production of double-stranded RNA. As you know, in eukaryotes, double-stranded RNA is not supported. So it gets fragmented. The fragments, the small fragments of that RNA is known as siRNA. And that siRNA has the capability to bind to the protein and make it inactivated, right? So the answer for this question is option A. Okay, next question for the day is, the process of RNA interference has been used to develop in the development of plant resistant to option A, nematodes, B, fungi, C, viruses, and D, insects. You don't require to take more than 10 seconds, because we have just discussed this question, that for the resistance of which particular kind of pathogen or an organism, right, we are using the RNA interference technique. Those who are unable to give the answer in poll can give their answer in chat box. Chat box is open for all. You have to give your answer. Okay. So everyone has given answer in chat box, that the option is A, right? That is nematode, right? We have initially developed this technique for the tobacco plant because a nematode parasite was existing in the roots of the tobacco, due to which there is deficiency of nutrient for the plant and leading to the decreased yield. Okay. So next we are moving towards our 14th question. The 14th question says, the Bacillus thuringicensis forms the protein crystals which contain the insecticidal proteins, and this protein, you have to find out the correct statement regarding this protein. Your option A says binds with epithelial cell of midgut of the insect pest in ultimately killing it. Option B says it's coding by several genes including the gene Cry. Option C says is activated by acid pH of the foregut of the insect pest, and option D says does not kill the carrier bacterium, which is itself resistant to this toxin. Okay, so we have discussed this concept several times in this chapter today itself, right? Because this question has arisen several times. This question has came several times. So, right, the answer you have given. Okay. So the Davangiri, you have given the answer. The answer is option A. We know here the Bt toxin crystals are formed here. And whenever the insect bites the plant, those crystals of Bt toxin enters inside the gut of the insect, and due to the presence of alkaline pH, those crystals get activated, or we can say the toxin get activated and cause the lysis of the midgut of the insect, due to which the insects dies because of starvation, right? Okay, let's move to the 15th question. The question says silencing of mRNA has been used in producing transgenic plants resistant to. Here we are saying we are silencing the mRNA. See, the same types of questions are appearing, but in a different format. So you have to be focused, be attentive while giving the answer, that the same question has appeared in a several manner. Option A, bollworm; B, nematode; C, white rust; and D, the bacterial blight. So we know the silencing of mRNA technique is RNAi technique, that is RNA interference. And this, right, Davangiri, the RNA interference technique is used for the parasitic nematodes which are found in the roots of the tobacco. Okay. So the next 16th question of the day, it says the genetically modified, or we can say the GM, brinjal in India has been developed for. Why we are developing the genetically modified crops? Specifically here, the brinjal is. Option A, insect resistance; B, enhanced self-life; C, enhancing mineral content; and D, drought resistant. Okay, so now you have to give your answer. Why we are developing this genetically modified brinjal in India? You have to give your answer in chat box. Okay. Very good. Okay, so as most of you have given your answer in the chat box, that it is option A, insect resistant. We want to increase the yield only by decreasing the damage caused by the insects. So for that, we are also not requiring the spraying of chemicals because they ultimately affect the humans, right? So for that, we are introduced the genetically modified plants, right? The next question, number 17, says some of the characteristics of Bt cottons are. Now we have to find out the characteristic of Bt cotton. Option A, long fiber resistant to aphid; B, medium yield, long fiber and resistant to beetle pest; option C, high yield and production of toxin protein crystals which kill diphtheran pest; option D says high yield and resistant to bollworm. Right, that is a correct answer. It is producing the toxic protein crystals which are able to kill the diphtheran pest. So yes, answer C is correct. Okay, so next question for today. An improved variety of transgenic basmati rice. Here we are talking about the transgenic basmati rice. The term transgenic itself means the genetically modified. Okay, please don't take tension, don't get confused. The transgenic and the genetic modified, both are the terms having the same meaning. So we are using the improved variety of GM basmati rice, or we can say the transgenic basmati rice. A, does not require chemical fertilizer and growth hormone. B, high yield and is rich in vitamin A. C, is completely resistant to all insect pests. And D, gives high yield but has no characteristic aroma. Okay, so give your answer. Everyone give your answer. What is the answer? Why we have made the genetically modified basmati rice? Okay. Yes. So answer is B. We have already studied. We have made the golden rice. Guys, that golden rice is also a basmati rice. Okay. So golden rice. Don't get confused. As I told you, don't get confused with the names. We are using several names for a same thing. So transgenic basmati rice, golden rice, both are same. And those are rich in vitamin A. Okay. So answer is vitamin B, that is gives high yield and rich in vitamin A. So the next question of the day, which is true about Bt toxin? You have to find the correct statement about Bt toxin. Option A says Bt protein exists as active toxin in the bacillus. B says the activated toxin enters the ovaries of the pest to sterile it and thus prevent its multiplication. Option C, the conquered bacillus has antitoxins. And option D says the inactive protoxin gets converted into the active form in the insect gut. Okay. So one of you have given the answer. The rest, please give your answer. The answer is required. Why we are using the Bt toxin? Okay. So as you all have given the answer and as we have already discussed this concept that the Bt toxin is an inactive protoxin which get converted into the active form only after getting the alkaline pH which is present in the insect gut. Good. Next question is this. So next question is saying transgenic plants are the ones. Transgenic plants are the ones. You have to find out the correct statement regarding the transgenic plants. Option A says generated by introducing foreign DNA into a cell and regenerating a plant from that cell. B, produced after protoplast fusion in artificial medium. C, grown in artificial medium after hybridizing in the field. D, produced by the somatic embryo in artificial medium. So the term is transgenic. Transgenic stands for the genetic modification. Yes, so you have given the correct answer also. So I'm very impressed, Davangiri, because you are giving continuously the correct answers. So keep on making the space. I'm definitely sure you will crack the need. Just keep your pace this much high. Okay. So yes, the answer is A. It is generated by introducing foreign DNA because we know the term transgenesis means genetic modification, right? So yes, we are regenerating a plant by making the recombinant DNA, and in recombinant DNA we always introduce a foreign DNA. Okay, so next question. The bacterium Bacillus thuringiensis is widely used in contemporary biology as. You have to give your answer. A, insecticide. B, agent for producing a dairy product. C, source for industrial enzyme. And D, indicator of water pollution. So give your answer. Sorry. Okay. So yes, you have given the correct answer. It is using as an insecticide which we are using broadly to generate the genetically modified crops, or we are commonly call them Bt crops, which are resistant to the insecticide. So your next question for day is, what is antisense technology? What is antisense technology? Option A, when a piece of RNA that is complementary in sequence is used to stop expression of a specific gene. B, the RNA polymerase producing DNA. C, a cell displaying a foreign antigen used for the synthesis of antigen. And D, the production of clonal variants in tissue culture. So you have to select your correct answer in chat box. JNV UK, you have to participate, beta. Otherwise, it would not be possible for you to analyze your answers, right? So please start giving your answers. Okay, so poll will end in next 10 seconds. Okay, I'm going to end the poll in the next 2 seconds. Okay. So yes, the answer is C. We know the antisense technology is used in RNA high technique, that is RNA interference technique, in which we are making the piece of RNA that is complementarily sequenced, which is used to stop the expression of a specific gene. Okay. So next question, 23rd. It says Cry1 endotoxin obtained from the Bacillus thuringiensis are effective against. Here we are talking about the Cry1. We know there are two types of gene, Cry1 AB, Cry1 AC, and Cry2 AB, right? So three genes are here, out of which we are talking about the Cry1. So yes, very good. The option B is the correct answer. Yes, that is used for killing, or it is used against the cotton bollworm. Okay. So your question number 24, it says, a transgenic food crop which may help in solving the problem of night blindness is developed in developing country is. In developing country, now we are using a genetically modified crop which is enriched in some nutrient which prevents the deficiency, or we can say which prevents the night blindness. And it is done by the process of biofortification, and by using the biofortification, we are producing, yes, the answer is correct. The answer is B. That is golden rice, because golden rice is introduced with beta carotene gene from a daffodil to increase the production of vitamin A. And we all know the vitamin A deficiency causes night blindness. So here we are preventing the night blindness. Okay. So the next question is, main objective of production or the use of herbicide resistant GM crop is to B, encourage eco-friendly herbicides. B, reduce herbicide accumulation in food articles for healthy safety. C, eliminates weed from the field without the use of manual labor. And D, eliminates weed from the field without the use of herbicide. So you have to give your answer. Okay. JNV UK, you have to also give your answer. Okay, perfect. Yes, the answer is C. As we have already discussed, the herbicide here used is glyphosate, which is disrupting the shikimic pathway, which is responsible for the production of growth hormone, that is auxin. Okay, so yes, we are eliminating this, or we are making the GM crops herbicide resistant, so that only weeds will get affected and our crop will not get any damage, and without using the manual labor we are able to destroy all the weeds. Next question. Golden rice is a promising transgenic crop. When released for cultivation, it will help in A, producing a petrol like fuel from rice, elevation of vitamin A deficiency, C, pest resistance, and D, herbicide tolerance. Very good. So everyone has given the answer. Yes, the answer is B, alleviation of the vitamin A deficiency. Okay, due to which we are able to cure the night blindness. Next question says the Bacillus thuringiensis, that is BT strain, have been used for designing novel A, biofertilizers, B, biometallurgic technique, C, biomineralization process, and C says the bioinsecticidal plants. So you have to give your answer. Out of these four, what type of plant we are producing? Okay, everyone. Nice. Yes, it is used for the development of bioinsecticidal plant. That means insect resistant plant. Okay, it is used for making the insect resistant plant. Good. Okay, so next question for the day. The golden rice is a transgenic crop of the future with the following improved trait. Example, option A, the insect resistance, B, high lysine content, C, high protein content, and D, high vitamin A content. Now you have to give the answer in the golden rice. We have improved the trait which is responsible for. Give your answer in chat box. Yes, correct. We are increasing the amount of vitamin A content. We know that the golden color of this rice is due to the introduction of beta carotene gene, which is responsible for the production of vitamin A. So yes, the golden rice is a rice which is having the enriched vitamin A content. Next question, question number 29, or the last second question of the day, is the first transgenic crop was. The first transgenic crop which we have formed was tobacco, cotton, pea, or flax. You have to give your answer. Right, the option is cotton. So BT cotton, which is formed, that was the first genetically engineered crop which we have introduced in our country. So the answer is BT cotton, or we can say the cotton itself. Okay, so the last question of the day, you have to select the correct. You have to select the correct column and the option. So column 1 has given some techniques and deficiencies, whereas column 2 is given their use or effect. So you have to find out which is a correct option. Okay, I think the options are not given here. We have to match the column A with column B. Okay, so you can give your answer. For A, the answer is what? B, answer is what? For C and for D, what are the correct answers? Okay, for A, that is BT cotton, we have the correct answer From the column B, first, second, third, or fourth, which one is correct? The BT question. The correct is fourth. That is Bacillus thuringiensis. Because the BT in the cotton itself stands for the Bacillus thuringiensis. Option B is adenosine deaminase deficiency. We have studied this deficiency in chapter 5. So for option B, okay, we have answer 1, right? The ADA deficiency is related with the first gene therapy which was done in a 4-year-old child, baby child in 1990. Okay, then RNAi. Yes, it is a cellular defense mechanism by inhibiting the function of protein. And the last, PCR. Yes, it is a technique used for the detection of HIV infection. Okay. So as you have given all the answers correct, very good to everyone. Okay. Please keep practicing like this. Keep making your concept strong. Okay. We'll meet tomorrow with the next chapter, right? So as we have completed our genetics, so from tomorrow onwards, we'll start with the ecology. Okay, the next high weightage unit. Okay, so be prepared for that. We'll meet tomorrow. And before leaving the class, please give me your feedback through this NPS poll. Thank you so much for your precious feedback. Okay, we'll meet tomorrow and you may now leave the class. Those who have given their feedback can leave the class, and the rest please give your feedback so that it helps in the improvement of class. Okay. Thank you so much. Have a good night. Okay, we'll meet tomorrow with a new chapter and the new Josh also. Okay, bye. Good night.